Buenos días. En el día de hoy, como parte de las charlas de los jueves de la universidad, tenemos invitado al doctor André KESSLER. El doctor KESSLER obtuvo su maestría en Ecología, Geobotánica y Genética de la Universidad de Würzburg en el año de 1998. Obtuvo su doctorado en Ecología en el Instituto Max Planck de la Universidad de Jena en el año 2002. Recibió la medalla Otto Hahn por investigación de excelencia en el año 2003. Este es un premio que otorga la Fundación Max Planck a los jóvenes investigadores que están iniciando carrera o que están trabajando en unos campos de investigación novedosos. Actualmente, o desde el año 2005, es profesor de la Universidad de Cornell en los Estados Unidos. Sus trabajos se han centrado fundamentalmente en la ecología molecular y química, en interacciones planta-insecto, en sistemas básicos y aplicados. Su trabajo de campo lo ha desarrollado en los estados de Nueva York y Utah en los Estados Unidos, además de Perú y Kenia. Y adelanta un proyecto para establecer un nuevo método ecológico y socioeconómico sostenible para el cultivo de cereales importantes. Tiene cerca de 35 publicaciones, 3 de ellas en la revista Science, en revistas como Ecology, el Ecology Lecture también, 5 capítulos de libro. Hace parte o ha hecho parte del comité editorial de Plant Phytology y de Ecology, y ha sido invitado y coordinador a las conferencias Gordon, que son eventos internacionales en los que se invita a los investigadores que tienen temas de relevancia internacional. La charla que nos va a dar hoy se titula The Information War in the Plant Space, o Guerra informativa desde las perspectivas de las plantas. Bienvenido, doctor.
Muchísimas gracias.
Gracias.
And first, my apologies, I will speak in English, not in Spanish. You may try to ask me something in Spanish later on, and I try to answer that, maybe in English then again. So I understand most of Spanish, but I'm so bad in speaking Spanish that it would take me 3 times longer to give this talk in Spanish than it does in English. The information war in the plant headspace, I chose a very provocative title for 2 different reasons, but before I give you the reasons, I want to give you a little introduction into what my lab is actually doing. And I should first actually say that I'm very thankful for being here, and in particular to Carlos and Marisol for hosting me here. And I had a wonderful time already. Unfortunately, it's already close to the end of my time. But the interesting thing is that everything that we do in my lab is seemingly merging very nicely into what's done here. And I hope that we can start some larger collaborations for the future. And I think they already have started. So what we do in my lab is overall study plant-induced responses to herbivory. That means, how do plants change their physiology in response to herbivore damage? And how does that change in the plant's phenotype affect back to the arthropod community associated with the plant, or whatever, the general community associated with the plant. So we have a large project on a wild tobacco plant, Nicotiana tenuata, that grows in the southwestern United States in the desert habitats of Utah. And there we study the specificity of these herbivore-induced plant responses, meaning, and I will talk a little bit about the specificity of plant responses today as well. What that means is, if 2 different herbivore species attack one plant, does the plant respond differently to the 2 different herbivore species? And if so, does that have effects on the arthropod community? The second large system that we work on is a solidago plant. So this is Nicotiana, this is solidago, which is a goldenrod. And you may know that the northeastern United States is actually the center of species diversity for this genus. It's one exception, right? Otherwise, the centers are usually here. And there we study ecological consequences of induced responses in general. It's a large study system. But what I want to talk about today is primarily what we do on wild tomato species in Peru. And you will hear more about that in a second. Now, when I talk about induced plant responses and its effects, and now I come already to that point of information war. You have to know pretty much what's on that picture is everything you have to know for understanding my talk today. So if an insect herbivore, like this caterpillar here, is attacking a plant, it does a certain amount of wounding, and different herbivores do different amounts of wounding and different kinds of wounding. But at the same time, these insects apply chemical elicitors. They might be just coming from the surface, from the cuticula of the insect, but in most cases when they're feeding, it comes from the regurgitant, from the foregut of the insect, or from the salivary glands, right? And these compounds are very different, and they alter, in general, wound responses to the different herbivores and cause the high specificity that we often see in some plant species. And so this damage actually is triggering plant endogenous signaling pathways, and some of them are relatively well known. So ethylene, abscisic acid, and auxin in general are known, from a basic plant physiology class. These are growth hormones, and growth hormones in general change in response to herbivory, the production of those growth hormones. So rapidly after herbivore damage, but the most important ones for a plant wound response are, in this group, the octadecanoid pathways that lead to the production of jasmonic acid, which is another phytohormone. And it was first identified as a growth inhibitor, but later it turned out that this hormone is the crucial hormone for mediating the plant's perception of wounding. So if you don't express that hormone, if the plant is not able to express that hormone, we did experiments like this, then the plant is not able to perceive damage, and you will not see any response of the plant to just mechanical damage and any kind of herbivore damage. So to make that short, there are many different signaling pathways that are triggered, and they're altered by the chemical elicitation coming from the foreguts or from the salivary secretions of the insects, and that leads to what we call a signaling pathway crosstalk in the plant that eventually changes the transcriptional levels of the plant, and that leads to a metabolic reconfiguration. And that, in turn, is pretty much what we can measure. So we can measure a chemical change, the change in secondary metabolite production in the plant in response to herbivore damage. Now, whether or not that change actually has an ecological effect is defined by that effect. So in many cases, the increased production of secondary metabolites leads to an increased resistance of plants that were previously damaged. And this is what this talk is all about. But secondly, there are some compounds that are not necessarily toxic or antidigestive and increase resistance, but they are emitted into the air. These so-called volatile organic compounds have multiple functions, and this is the focus of the talk today. So we want to talk about these volatile organic compounds, odor compound smell, that is emitted into the air in response to herbivore damage. Now I give you one example from our work on wild tobacco, and there's a reason why I give you this particular example. I come back to it in a second. So wild tobacco plants, when attacked by insect herbivores, and the most important herbivore is a sphingid caterpillar called Manduca quintimaculata. And when they attack the plant is emitting volatile organic compounds into the air. In this particular case, there's a predaceous, a predator bug, a Geocorida bug, Geocorus pullens, that is using these volatile cues to find their prey, which are these caterpillars, right? So the plant, you can say, is actually crying for help. Right? And that's why people call that kind of mechanism indirect resistance, because the attraction of that predator to the plant and the more efficient pretty much prey search of that predator increases the fitness of the plant, or can theoretically increase the fitness of the plant. Interestingly, if you take the adult moth, so this is Manduca quintimaculata mama, the adult moths use that same volatile signal to avoid ovipositing on those plants. And there are two important reasons for that. First, if they lay their eggs on a plant that is already crying for help, the predator will be right there and kill the hatching caterpillar. It will actually kill the eggs already, right? So there's a very strong protection in the plant by those predators when the plant's already attacked. But secondly, this volatile emission is just representing a general metabolic change in the plant. So there is, in fact, an increased production of nicotine, a toxic compound, right, an insecticide, if you will, of proteinase inhibitors, which are anti-digestive compounds, so they make the insect not digest the proteins. So all those compounds and a series of other compounds are upregulated in response to damage. And the volatile emission is just one part of that metabolic change. So that means the adult moths not only avoid predation pressure, but also bad food quality, right? And we actually calculated from the experiments that we did in the field that the plant's ability, and we did actually an experiment where we manipulated by genetically transforming those plants not to be able to emit volatiles. So we tested that actually directly. Whether or not, so plants that are not able to emit those volatile compounds in response to damage have a 90% higher chance to be damaged, right? Or turn it the other way around, plants that are able to emit those volatile compounds reduce the herbivore load on them by 90%, which is better than an insecticide, right? It's a very special case, and that suggested that we have a high fitness in those plants. Nevertheless, there's a big fitness benefit in the plants, right? Nevertheless, there is this big question in the field whether or not the predators can actually be the agent of natural selection on those volatile traits. And this is actually pretty much the information war that I want to talk about, that one part of the information war. Because in the field of studying plant volatiles, and especially herbivore-induced plant volatile emission that we see here, it was assumed that this inducible volatile emission is in fact has evolved in order to attract predators, to make it really straightforward. But now, I sat together with a friend of mine and we looked into the literature that's actually supporting that. And we looked into all the literature that's looking into what we call indirect resistance traits. And as you can see, you can sort indirect resistance along continuums. On one hand, we have ant-plant-ant interactions that are very common here in the tropics. So this is an acacia plant interacting with ants. And in this particular case, it's an obligate ant-plant-ant action. So the ants cannot live without the tree, and the tree cannot live without the ants. In this case, the plant is providing food and shelter. And then we go to the opposite. Again, here we have Geochorus pollens from the tobacco example. In this particular case, we have only volatile organic compound production. And we could say that this also represents a continuum from totally obligate, and there's a continuum already in here because you know there are facultative ant plants as well, to totally facultative, because who is responding to that signal is very open. And we call that, in order to differentiate between those two extreme types, we call them resource-mediated indirect resistance traits and information-mediated indirect resistance traits. So in this case, the plant is only providing information. Here's an insect herbivore feeding. In this particular case, the plant keeps the predator around at all times and feeds the predator and keeps him home and everything, right? Now, if you compare those two with each other, there's some very interesting evolutionary thoughts behind that. So first, in this particular case, we have a lot of evidence in literature that we have actually direct selection of the predators on those traits, meaning food and shelter, right? So there's a lot of evidence for that. Direct selection. We have usually a high intimacy of the interaction, although I have to say there's obligate versus facultative, so there's a continuum again in there. And so we actually have in the literature evidence that the predators in this case, helping the plant, are the agents of natural selection on the traits that mediate the interaction, which are the resources, right? Food and shelter. Now, if we go to information-mediated indirect resistance, we have a more indirect selection, and I will tell you in a second what I mean by that. We have a low intimacy because the signal is not made for that particular predator. It could be any predator that can read the signal, right? And the question is it actually made for the predator? Because we had to conclude there's absolutely zero study that shows us that a predator can be the agent of natural selection on the volatile traits, right? So there is no study. And the closest study we come is actually our study on wild tobacco, because we actually measured fitness effects on plants that were not able to produce volatiles in the field. And so there we may have it, but it could be a very special case. Now, what do I mean with more indirect selection? So we can turn that kind of question for what does that mean around and ask, what does it need for a predator to be the agent of natural selection on those volatile traits? First, there is a volatile cue that must attract a natural enemy and facilitate prey search behavior. And the example that I gave you was a good one, and we have at least 32 other plant species in which that works perfectly fine. So this is okay. Predators go towards a volatile signal and they usually kill the herbivores. The second thing is the herbivore inducing the volatile cue has to have a negative fitness effect on the plant. That sounds trivial, right? Herbivores are always bad, or are they? Right? So this is the problem, that if you work with plants and herbivores, you often find that the herbivore doesn't have any fitness effect on the plant. There's only some cases where there are very strong herbivores that reduce actually seed sac in a plant. And so in many cases we have a lot of herbivores that don't have significant fitness effects on the plants. In which case a predator coming killing that herbivore has also no fitness effect, right? And so in third, you need a predator, this is pretty much what I just said, attracted to those volatiles that reduces the herbivore performance of a herbivore that has a high fitness effect on the plant. And this is what I mean with a more indirect selection on that trait. Now we had to conclude from that study that it is actually relatively unlikely that the predators are the actual agents of selection on those traits. And this is even more pronounced when you look into what does it mean. So again, this is the study that it comes the closest to it, but it may be a special case, right? But now, when we look into what other functions of volatile emission have been found, of inducible volatile emission. So first we have indirect resistance, predator attraction, which was the oldest. But meanwhile, we know these volatiles mediate plant-plant interactions. So a plant emitting volatiles can in fact induce resistance in a neighboring plant, right? Or within a tree, a branch damaged by the herbivore on this side of the tree can, via the volatile emission, induce resistance in a distant part of the tree without using the vascular system. So more efficient transmission of information within a modular plant, right? And in this particular case, the selection on the trait would be direct because it's in that plant. The other thing is herbivores directly select on that trait because they use that volatile cue as a host finding signal. So whether or not a herbivore is avoiding an emitting plant or is attracted to an emitting plant, it will be a direct agent of selection on that trait if it has a fitness effect on the plant. Now this part here is what I want to focus on today. And I want to use special herbivores, namely pollinators. Pollinators are herbivores as well. They have to cope with the same plant secondary metabolites as the herbivores do on the particular plants. Right? And this is actually a study that we accidentally fell into because when we started working on wild tomatoes, we had a totally different idea about what we actually want to do. But because nothing, absolutely nothing was known about the ecology of wild tomato plants, except where they're growing, we started general surveys of herbivory on the plants, pollination success, and these kinds of things. And what we found first, and this is in Peru, nice Peru, very high up in the Andes, and this is actually where the tomatoes grow in the Andes, and these are so-called seasonal triforests. There are not many trees there. This is the Pacific slope of the Andes, extremely dry habitats, and all so far described 19 species of tomatoes, wild tomatoes, are growing in these very extreme habitats, right? And they are usually the only plants that flower in the dry season. And the dry season there is something different from the dry season here. This is extremely dry. The people who were involved in this study were, very importantly, my wife, Katja Poveda, who some of you know here, because she did her master's thesis here at the University of Bogota. And Rai Kalichko is a postdoc in my lab, was very important in the study. And just more recently, another postdoc in my lab, Mark Severin. Now, the study that we go first to, so I have two different studies that I show you. The first one is a little bit north of Lima, in a village called Cajacay, close to the village of Cajacay. And this is how the habitat looks like, and this is where the tomatoes grow. These are actually tomatoes, you can barely see it. And this is how they look like. This is one wild tomato species, one of the most widely distributed wild tomatoes, called Solanum peruvianum. Solanum peruvianum. It's actually better in Spanish and in German for me too. What we found in the survey was that there's a large number of herbivore species associated with the plant, and these are just the most important ones. There's another sphinxid moth that is very common there. Most important herbivores are flea beetles, usually Epitrix, genus Epitrix flea beetles, and there's some more Lepidoptera species, and this is just a Spodoptera as a representative. And then we already did tests in the lab with these plants, and we knew that we have herbivore-induced secondary metabolite production and induced resistance to herbivory. So a number of compounds are upregulated in response to damage, and that results in an increased resistance to those herbivores. So we knew that already. What we didn't know was that the pollinator community in this plant is very diverse, and these are just three representing the large groups that are pollinators on these plants. And these were colitid bees, apid bees, so primarily the genus Xylocopa, and halictid bees. And there were several species in those, and 3 species of Xylocopa. What's important to know is, you see that already here, all the bees that actually visit those flowers are specialized in a way. They're behaviorally specialized. Solanum flowers don't give off any reward easily. They don't have nectar, but they have very good protein-rich pollen. But that pollen you only get as a bee when you hang onto the flower like this and vibrate your wings in a certain frequency. The pollen shoots out and you clean it off and carry it home. So these are all behaviorally specialized bees. Honeybees cannot pollinate a solanum flower, right, for example. Now, when we did the survey, the first very important pattern was that we compared overall herbivore damage, and this is a very crude herbivore damage measurement. This is the percentage of leaves on a particular branch damaged, right? So from 0 to 100% damage, and that means any type of leaf. So we counted a leaf damaged when there was a little point. So a lot of that is actually damage that is below 10%, even in that range here. So that means any leaf damaged, the percentage of the number of leaves damaged, right? And at the same time, we measured the proportion of flowers with pollination marks. What are pollination marks? Look down here. So when the bees hang onto the plant, they have to grab very tightly when they vibrate their wings. And in that situation, they damage the plant slightly, and that leads to a reaction on the surface of the tissue that leads to a browning of the tissue. That appears within 5 minutes after the bee has visited. And it will only appear when they actually try to vibrate their wings, because then they had to hold on. So this is a very secure sign that there was a pollination event happening, right? Whether or not it was successful or not is not important, or at least is not, or we cannot say it's not important, it's wrong. We cannot say, but it's very important for the plant, by the way. It is definitely a visiting and reflecting pollination success. But what you see here is the proportion of damage is strongly negatively correlated with the percentage of flowers with pollination marks. And that kind of negative correlation is also reflected when we look into the fruit production per inflorescence. You see also that negative correlation. Well, it's only a correlation. There are 2 possibilities why that could happen. First, the question is there actually a causal link between herbivory and pollination? In which case, it could be pollinator limitation, induced pollinator limitation. So herbivory pretty much repels herbivores from the plant. And the second possibility is herbivory reduces the amount of resources that can be allocated into food and flower production, right? So these 2 possibilities are there. Now we started to look into whether or not there is a causal link. So we did experiments where we had plants that were undamaged, and we applied damage, in this particular case Epitrix beetles, onto one plant and looked how the pollinators accept those plants. and in the others we had no herbivore damage. And what you can see, you see that same pattern. Again, this is the proportion of flowers with pollination marks and a relatively strong reduction. And I have to say that in all of the cases where we used flea beetles here with damage, we had less than 10% damage, much less than 10%. So it's almost nothing on a tomato plant. You know, people who know tomatoes, tomatoes are probably the most herbivore-tolerant plants you can know. Domesticated tomato, you can remove 50% of the leaves, you will get the same fruit set and the same seed set. They're very tolerant, and it's true for the wild species too. Now, interestingly, when you apply methyl jasmonate, remember I told you about this phytohormone. This is important in wound response, right? Jasmonic acid. This is the methyl ester of jasmonic acid. When you apply that to the plant, you mimic herbivory without any damage, right? And that actually results also in that response, meaning that this response to the pollinators is mediated by mechanical damage in the plant, right? And that already suggests that it may be something that has to do with the attraction of the pollinator rather than with the allocation of resources, because here we don't cut any tissue away, right? But we had to test that in more detail. What we did was, go into fruit production. Does that actually change it? That reduced attraction of pollinators, does it change fruit production? And in fact, it does. So this is now the number of seeds per fruit for flowers that we followed through all the way to seed set, right? So we had the treatment in the beginning, beetle damage, saw whether or not the pollinators were coming, and then followed those flowers through, and you see this reduced seed set per fruit. Now when we apply methyl jasmonate, we see exactly that same pattern, right? Now if we assume pollinators are not important, so we hand pollinate, this is what we did here, we just hand pollinated all flowers in the experiment, we actually get a slightly increased production with methyl jasmonate. And that actually shows you it's not a problem of resource allocation, but it is in fact induced pollinator limitation. Herbivore-induced pollinator limitation, this is how we call it in the end. Right, now the question is, why do the bees avoid flowers on damaged plants? You can ask that question from a functional perspective and from a mechanistic perspective. From the functional perspective, I can only speculate, and I give you some sort of hints. On the functional level, the first hypothesis is there may be a correlation between the fall secondary metabolite production and the pollinator reward quality. So remember, we know in response to herbivory, plant is changing the secondary metabolite production. We already know that we have induced resistance. Maybe the pollen as a reward is also bad, right? Has changed chemistry. Now we looked into that. First we saw actually very strong correlation in some of the compounds that have been identified in other systems as defensive compounds. Chlorogenic acid is a phenolic compound, phenylpropanoid, rutin is a flavonoid, but also phenolic compound in that sense. In both of them we find that the leaf concentration is strongly correlated with the concentration of the compound in pollen. Now if we take, however, the entire chromatogram, so these are liquid chromatograms, each of the peaks represents, ideally represents one compound, if our separation was good. What we see here is that there is already something very different in the leaves versus in the anthers. So there are some compounds in the leaf that we don't find in the anther cones, that we don't find in the pollen. And then there's a whole group of compounds that we don't find in the leaves, but we find them in the anther and the pollen, right? And these here are actually coumaric acid derivatives, and I just put coumaryl here as an example for that. Also phenolic compounds, and also suspicious of having defensive functions. So we have actually both, and not a clear answer to that. We have a strong correlation in some compounds, but we also have a very strong separation here. And I come back to that later. The second hypothesis is that herbivory-induced volatile emission may actually increase, or actually the presence of the herbivore on the plant may increase the number of predators on the plant, and that may be a danger for bees as well, right? And interestingly, we see a correlation between damage and the presence of actively hunting spiders, like jumping spiders here or crab spiders. So there is an increased number of those spiders around, but we don't have a causal link there, it's only correlation. Now the third hypothesis is the one that we actually favor the most, and we have a few bit of evidence for that, and this is the constancy behavior, meaning that bees are constant on a particular type of flower, and they may use whatever information is changing in response to delivery to avoid flowers on the plants, meaning they pretty much perceive the flowers as something different than what they usually had. And we can address that a little bit more in detail and go more into the mechanism. Because my lab studies primarily these volatile organic compounds, we started with comparing volatile emissions from flowers on damaged plants and flowers on undamaged plants. And the way we do it, some of you know now how we do that. We enclose whatever tissue we want to trap from into a cup, and then pull air with a vacuum pump over a charcoal trap that holds onto the organic compounds. We then elute down those organic compounds with dichloromethane and analyze them in a gas chromatograph and mass spectrometer, right? And this is how that looks like in real life. And you see often it can be very dangerous. There's steep mountains in Peru. This is Ecology, my postdoc. And this is actually not the trapping here. These are the covers that we use. These are sleeve bags that we use to manipulate herbivory, right? So what are the results? This looks crazy. I mean, these are now gas chromatograms, not liquid chromatograms, but still every peak is representing one compound ideally, right? In some cases you see that there's not a good separation. This peak is probably 3 compounds. What's important, and I want to focus your attention first on the upper 2. So these are leaves on plants that were not damaged, and you know tomato leaves are already aromatic, they smell without any damage, right? So we have a lot of peaks already in the undamaged leaves, but when we have damage, and in this case by a Manduka caterpillar, we get a strong induction of a large number of compounds, primarily in front here, monoterpenes, and back here, sesquiterpene compounds, right? So you have a large number of compounds that are induced in response to herbivore damage in the leaves. And this is kind of expected. We almost always find that in different plant species. Now, interestingly, there are compounds that are only produced in the flowers and are not appearing in the leaves. This was also not necessarily a surprise, but to us it was a surprise because for us, tomato flowers don't smell different from the leaves, right? And so that's why it was a surprise to me, because I couldn't definitely not smell a difference there. But obviously there are differences, but the big surprise was that we actually see compounds that are only produced in the flowers and only in response to herbivore damage. So that means herbivore damage on a leaf is systemically changing volatile emission in the flowers. And so now we have a potential signal that the pollinators actually could use, the bees could use, and maybe avoid plants on damaged plants over undamaged plants. Now, in order to test that causally, there's one problem. We couldn't take the bees home to Cornell because of restrictions, and most of the bees were solitary bees, and so we couldn't do good experiments anywhere else than in Peru itself and in the field. And so we had to come up with a choice experiment that we can use in the field. Now, we could take advantage of the fact that the response in those plants is independent in 2 different branches. So if you take one branch and damage this branch on the plant, you will not measure an effect in the other branch. So that gives you the possibility to have an inflorescence, the flowers, on a damaged branch and bring them together with flowers from an undamaged branch into one inflorescence. This is what that picture is about here. So if it is a visual cue that the bees use in order to differentiate between damaged and undamaged, they would still differentiate, right? We would still get more pollination in the ones that are undamaged and not in the ones that are damaged. If it's an olfactory cue, they will not be able to differentiate. And in order to test that even further, we did something like this. So enclosed the flowers on the damaged plants and exposed the bees to the visual cue of the undamaged. So now they have, it smells like damaged, but it looks like undamaged, right? And then you have to have the controls for it, right? And this is how that looks like. So this is the first experiment. In this particular case, we did that with herbivory as well, but in this particular case, we used methyl jasmonate to standardize the amount of damage. And so this is the open one, and there's always a control inflorescence. This is on undamaged branch, and in this case it's also undamaged because it's the control treatment. And here we have, this is an undamaged one, and this is the damaged one on the same one, and both of them get a reduction. And when you cover the damaged one, so both of those are undamaged, but here this one smells damaged, you get also the reduction. And this clearly shows in a field behavioral experiment that it's an olfactory cue that the bees use to avoid flowers on damaged plants. Right? Now we use this design here to test what part of the emission do they use. So on the flowers you also have green tissue, right? So it could be volatiles coming from the green tissue, and it could be volatiles coming from the corolla of the flowers. And what we haven't excluded here, sorry, to go back. What we haven't excluded here is that, here we go. What we haven't excluded here is that methyl jasmonate, which is also volatile, is repellent to the bees, right? So we have to test that too. And so we use that design. This is pretty much the same. This here is exactly the same treatment as this here. It's good we can repeat the same experiment again. In this particular case, we had damaged leaves in this leaf bag here instead of flowers, and there's no difference. So meaning that the volatile emission that changes the behavior of the bees is actually volatiles coming from the corolla of from the flowers rather than from the green tissue of the flowers, right? Rather than from the leaves. And here, luckily, methyl jasmonate doesn't make any difference when you put it alone into the bag, and so we were fine there. So what that says is herbivores choose and are able to differentiate between damaged and undamaged plants based on the volatile emission coming from the corolla of the flowers. Good. This is a short review of what we just talked about, and then I go into another short study. So we have induced resistance to herbivory, and we have an herbivore-induced pollinator limitation. And this is the first study that actually showed that, this kind of effect. The pollinators use the volatile cue during their foraging for pollen, and this results in seemingly ecological cost-induced resistance, which has never been shown before, and more specifically in an ecological cost of volatile emission, which also hasn't been shown before. And that made this study relatively special. This is a friend of ours, Katja. One day we were out in the fields and looking at our plants, and then there came a large group of 40 farmers down the road. And most of the tomato plants that we worked with were alongside the road. And he was kind of the boss of all the farmers in the village. And so they came down the road, and Katja explained them in a 1-hour talk what we are doing. And then he said, "Why are you doing all these experiments out here? Come closer to the village. I give you my fields and you can work there." And so we actually had common garden experiments in his fields. And so this is what actually interacting with the people helps the science. Now, we want to go into another system. So the data that I presented to you suggested that there is a very strong interaction between herbivory, the selection of herbivores on plant traits, versus the selection of pollinators on plant traits. And I'm talking primarily about chemical plant traits, right? And so meaning that there is a correlation, suggested correlation between reproductive strategy and resistance strategy of the plant, defensive strategy of the plant. So what do I mean with that? With reproductive strategy, I mean, for example, self-incompatibility and self-compatibility that should select for different defensive strategies if there is an interaction via plant secondary metabolite production. Now, there are three principal ways that we can already derive partially from what I told you in which plants could avoid that conflicting interaction, evolutionary. First, a plant could have flexible reproductive strategies. Second, there could be a separation of leaf and flower secondary metabolite production, and there was some indication of that in the example that I gave you. And the induced production of secondary metabolites may already be such a cost-saving strategy, right? And I want to show you some that goes into that. So when we brought up this kind of hypothetical framework In 2009, I wrote a paper about that. I had no idea what I was talking about in that paper, and there were no data available. But then there came a new PhD student along, Stuart Campbell, and he said, I want to test this hypothesis thoroughly. And he did an experiment that just blew my mind. So he took 58 species in the Solanaceous family, 34 of them self-compatible, 24 self-incompatible, in groups that were congeneric pairs, meaning that we always have, in one genus, for example, Petunia, we have one self-compatible and maybe one or two self-incompatible. In a genus Solanum, we have several because there are different clades in Solanum, self-compatible, self-incompatible. Then he did to each one of those species 3 treatments: a control, no damage, a mechanical damage treatment, and a Manduka treatment, and then fed those tissues back to the Manduka caterpillars, measuring resistance, induced resistance, and constitutive resistance. So we have measures for constitutive resistance traits, inducible resistance, and for the specificity of the induction, meaning is there a difference between mechanical damage and herbivore damage, right? On a phylogenetic map and phylogenetically controlled. The first thing that he found was actually disappointing. There is no principal difference in constitutive resistance between self-compatible and self-incompatible species. This is pretty much just as a weird picture, but there is no difference, I can tell you. But when he looked into the trait inducibility, you see already there is a shift, the blue away from the green. And when you put that together in one graph, you get this amazing and mind-blowing picture, for my opinion. Self-compatible species are, in average, more inducible in their resistance than self-incompatible species. And this is a very, very strong pattern. There's only 2 pairs in which it was not significantly different in the direct comparison here. And that results in an extremely highly significant value here, phylogenetically controlled, right? What you have to know in the Solanacean, and like in many other groups, self-incompatible is the original state and self-compatible is derived evolutionary. And there is no reversal of self-compatibility. So once you become self-compatible, there is no way back. When a lineage becomes self-compatible, there's no way back. And this is important because that has very strong indications on how to interpret those data. What that means is, in fact, that the evolutionary shift from self-incompatible to self-compatible somehow creates a selective pressure for increasing inducibility of resistance, right? And there's some other very interesting patterns that come out of that. When you plot inducibility to Manduka sexta against the inducibility to mechanical wounding, which is pretty much the question for how specific is the response. If there's no specificity, you have a very strong correlation. And this is what you see with the self-incompatible species. It's a very strong correlation between inducibility here, so meaning no specificity, while there's no correlation with the self-compatible ones, even though there are more species in there. So we're very confident that this means that self-compatible species are much more specific in their response to herbivory, and they're stronger responding as well. And in addition, we also found this long hypothesized relationship between constitutive resistance and inducibility. And this is, there was kind of costs associated with one or the other would result in a trade-off between the two, and there's a very strong trade-off, but this is obvious in both self-compatible and self-incompatible species. Now, to go to an example very quickly of wild tomatoes of 2 different species where we have kind of a case study of something like that. We go a little bit south of Lima, again in Peru. It's almost the same habitat, a little bit lower down in elevation, and there are 2 different wild tomato species, Solanum schmelewski, Solanum nericki. They're sister species, so they're very closely related, they're genetically almost identical. They're morphologically almost identical, except for the flowers, and I will tell you some in a second about that. And they grow in exactly the same habitat, they sometimes grow into each other. But they never hybridize in the field, although you can do that very easily in the lab. And the reason for that is one of them, Solanum schmelewski, It's a facultatively outcrossing species. It shows induced production of secondary metabolites, also induced resistance. It's a very strong induced resistance, in fact. And the other one is a sulfur. And you can actually see, here I actually covered it. So here the pistil is hanging out of the omphicone, and here it's totally inserted. So even if there was a pollinator coming, it would self in all of the cases. But this has also induced secondary metabolite production, but it has a higher constitutive resistance than this plant. Now when we look into the relationship between damage level and mean relative pollination, first we don't have pollination in the sulfur. That's why we don't have this picture for the sulfur, but this is for Solanum schmielewski. We see the same pattern that we saw in Solanum peruvianum, these other species that I told you about. And this is also actually causally linked. We also see induced pollinator limitation in this particular case. Now the interesting thing, the effect on seed number is again also negative, but you don't see any effects of herbivory in the sulfur, right? Again, suggesting it's not a resource allocation problem, it is in fact a pollinator limitation problem. Now, interestingly, when we measured floral volatile emission from those two different types of flowers, it looks actually extremely similar. Here it's a little bit higher because the flowers are bigger. So this is the outcrossing species, this is the sulfic species. So it looks almost identical, and if you average it, there's no difference except for one compound where there's methyl isoeugenol. And this is something that's extremely rare in chemical ecology, that you have one compound that explains almost everything. And I tell you what this compound is explaining. There's interestingly an induced emission of this methyl isoeugenol production that is reduced in response to herbivory in Solanum schmielewski. So there's one compound in response to herbivory reduces in emission, while there's nothing of that compound in Solanum neoricki. Now, we thought, why don't we make the test, how important is that one compound in fact? And use the same kind of design that I showed you before, and we manipulate the scent, but give them a different visual cue. So we pretty much have Solanum schmielewski and add a compound that is reflecting damage, which is beta-myrcene in this particular case, suggesting that we have reduced attraction of pollinators in response to an increased production of a compound that is reflecting damage, right? On the other hand, in Solanum neoricki, we have two ways of manipulating the volatile emission. Now, we don't have that one compound in the floral orders, so we can actually spike these flowers with the methyl isoeugenol, making that flower smell exactly like Solanum schmielewski, right? And then we can add beta-myrcene to it, like in the same experiment here, and see whether or not we get a reduced attraction of the pollinators. And the results were actually really staggering. So this is Solanum schmielewski, the outcrossing species, and these are the two different colors, are just two different days of measurement. And again, we measure the proportion of flowers with pollination marks, and you get a strong reduction with the emission of this monoterpene beta-myrcene reflecting herbivore damage. So again, volatile emission is explaining pollinator behavior. Now, this is the big deal. Here, lanolin is the actual control. In the control plants, there's no attraction of pollinators at all. We only find any kind of attraction if we add methyl isoeugenol, that one compound that only this species is producing. So we can pretty much manipulate the pollinators into coming to a flower that is usually not attractive to them. And we can reverse that by adding this beta-myrcene again to the plant, right? So meaning that pretty much everything that we observe in the behavior of the pollinators in association with that plant is mediated by volatile compounds, which was a surprise to us as well. Okay, now the question is still, why do they avoid those? And this is my last piece of data now. And there's this interesting effect, and I mentioned before, inducibility may be a way of getting around the compromising effects. Now what we see here, we have in the selfing plant, constitutively, and this is now on the same scale here, the production of a number of phenolic compounds is constitutively much higher than here. So these are constitutive levels. The white bars are constitutive levels, the black bars are induced levels in response to damage. So only when we have herbivore damage, we get an increase in secondary metabolite production in these in Solanum schmielewski, but we don't see that difference here. So that means by not relying on pollinators, you can view it this way, not relying on pollinators, these plants can defend themselves much better and their flowers much better. And the reason for why that is important, there are leaf colorons in this area that actually cut off the entire anthocone and pretty much resulting in zero fitness of the plant, right? And these are much better defended against those leaf colorons than they are. And so, on the other hand, if they were probably, so if these plants had a considerably high production like those, they would not be able to attract pollinators, right? By inducing the resistance and only being inducible, they overcome that problem. So all the time when they don't have herbivores, they can attract pollinators perfectly well, right? And so, and this is just an hypothesis. Now, with that, I want to go over this and then go to the conclusion, because I'm late already. So pollinators use volatile organic compounds when foraging. There's differential volatile emission that's affecting pollinator attraction. Plant volatile signaling mediates the interaction between pollinators and herbivores, actually, via the plant. We have a herbivore-induced pollinator limitation with significant plant fitness consequences. Herbivore-induced resistance, tissue-specific biosynthesis of secondary metabolites, and altered reproductive mode can be potential strategies of the plant, and these are hypotheses for future research to overcome the conflicting selection by herbivores and pollinators. And there's a core evolutionary process, and I think the data that I showed you showed it very clearly between plant resistance and reproductive strategy. And with that, I thank all the people that were involved in the study, and I hope we have some time for questions. I'm sorry I went so long. Thank you very much.
Tenemos 10 minutos para preguntas.
So, do you think that the tomatoes, the selfing, is getting the evolutionary response to herbivore because they can produce more chemicals? But they are getting high endogamy, so they will low ability to respond to new herbivores, to respond. So, is it kind of immediate response, but in a long time, how do we, or what do you think about it?
This is a very good suggestion. So the way I presented it is only one hypothesis, right? So this is a potential one hypothesis. It could be, the entire thing could be, in fact, and this is kind of what I wanted to show, a result of selection by pollinators, right? So if you have pollinator limitation, they're forced into selfing, they can free up those resources and can be better defended as a result of that. And so you can view it exactly the other way around. So it's not necessarily the way that high herbivory selected for selfing in this particular case, but it allowed to actually maximize the output of those plants that made it through that genetic bottleneck. Right? And you're right, they would not be able to respond to new herbivory, but this is kind of the question: is the habitat pretty much a habitat of high herbivory, relatively spoken, or low herbivory, and is the herbivory variable over time scales? And we don't know that. So but it's a very good suggestion.
¿Otra pregunta? Muchas gracias.
Muchas gracias.
Y a todos ustedes, muchas gracias por su asistencia.